Let's start by congratulations. It's been quite a week for you, I imagine.
Yeah, it has. Thank you.
The Nobel Assembly at Karolinska Institutet has today decided to award the 2024 Nobel Prize in Physiology or Medicine jointly to Victor Ambros and Gary Ruvkun.
When I spoke to Victor Ambros, he'd been a Nobel laureate for just a few days, and he was still smiling. How do you feel a few days out?
I feel good, and there's been a lot of surprises. This is the kind of thing that one doesn't really expect to happen. And so there's no planning for it, and it's not something you train for, so I'd say it's an adventure.
Yeah. What was it like waking up and finding out you'd won? I understand it was your son who actually got through to you, first of all.
Yeah, because my phone was off, and so somebody got a hold of my son, and I guess they started calling everybody by name of Ambros, maybe. And
Nice.
And then my son called my wife. So that's how we got the news.
Ambros and his co-laureate Gary Ruvkun were awarded the Nobel Prize for their discovery of MicroRNA, tiny molecules that tell the cells in our body what kind of cell to be. In this episode, we explore how these tiny controllers work to turn on and off bits of our DNA in a process called gene expression. And we hear about the potential of microRNA to open up new forms of treatment for cancer and other diseases. I'm Gemma Ware, and this is The Conversation Weekly, the world explained by experts. To help understand the science behind the 2024 Nobel Prize for Physiology or Medicine, I'm joined by Associate Health and Biomedicine Editor at The Conversation in the US, Vivian Lam. Hi, Vivian. Welcome back to the podcast.
Thanks for having me.
Vivian, I know you were excited when this year's Nobel Prize for Medicine was announced for the discovery of microRNA, because it's something that you've been covering in recent months. Can you tell us in very simple terms, what is microRNA?
So to understand what microRNA is, we can take a few steps back and consider what RNA is first. So RNA, like DNA, is a form of genetic material. So while DNA contains a so-called master blueprint of all your genes, RNA is a copy of that blueprint that cells actually use to translate those coded genes into proteins. Usually, when we talk about coding genes, it's like each gene contains a recipe to make a specific protein, and all your DNA contains different stretches of genes that have all the recipes to make up all the things a cell needs. So microRNA, as the name implies, is a specific type of RNA, but doesn't code for any particular protein at all. Rather, these short stretches of genetic material regulate gene expression. In other words, they control which genes are turned on or off by binding to and silencing the RNA that does code for proteins.
So there's these tiny controllers that control what every cell in our body actually does.
Yeah, pretty much. There's many different ways that cells can regulate how genes are expressed, and this is one of them.
Okay. And people may have heard of mRNA or messenger RNA, which was used to help deliver some of the COVID vaccines. But microRNA is different, isn't it?
Yeah, so mRNA, as you've likely surmised, is another type of RNA. There's a lot of types of RNA, and coincidentally, it was the subject of last year's Nobel Prize. It's a bit of a trend that's going on, it seems. So it's that copy of the DNA blueprint that I mentioned earlier that gets translated into proteins. So mRNA is what microRNA binds to when it turns genes on and off.
Okay. And the big question behind all this is something you just mentioned, gene expression. Why is that so fundamental to our understanding of biology itself?
Just because you have a specific gene doesn't necessarily mean it'll be active all the time or even at all. So which genes are expressed or turned on determine what proteins a cell produces and how a cell functions and interacts with its surrounding environment. And in turn, this all plays a big part in distinguishing, for example, like one type of cell from another, like liver cells and brain cells and skin cells, and from health and disease.
Great. Thank you so much, Vivian, for giving us that basic explanation so we can really understand what Victor Ambros is about to tell us.
My pleasure.
Today, Victor Ambros is a professor of molecular medicine and the Silverman Chair in Natural Sciences at the University of Massachusetts Chan Medical School. But the research that won him the Nobel Prize was published more than 30 years ago in 1993, when he'd just established his own research lab at Harvard University. Victor was trying to understand gene expression, the way cells get the right instructions from DNA during the developmental process.
It had already been understood that underlying development of an animal are changes in gene expression as cells, differentiate, divide.
Victor was exploring how this might happen by studying particular gene mutations, and he was using a special type of worm called C. elegans to do it. Is there something about these C. elegans worms, which are also called nematodes, that allows you to work with their genetic material more easily? Why are they so key to this type of science?
Yeah, I mean, as you may know, the C. elegans was developed as an experimental organism by Sidney Brenner in the 1960s. And Brenner was quite a visionary. He wanted to establish an experimental organism that people could use easily to first identify mutants and then study the development. And he also wanted to study the nervous system of this animal, the wiring of it. And so he looked for an animal that was simple, but still had all the various parts that are important to all animals, intestine, skin, muscles, a brain, sensory systems and behavior, complex behavior. So it only has about 1,000 cells, and all those cells can be seen easily through a microscope in the living animal. And in fact, you can watch all the cells as the animal develops. And that was a major attribute of this system because John Sulston, another Nobel laureate, and Bob Horvitz produced a map of the development of this animal. It's called the cell lineage, and it's reproducible from animal to animal to animal. So it's quite amazing system to allow investigators to study developmental processes and mechanisms really on the level of individual cells and what those cells do as they divide, and differentiate during development.
It's also pretty easy to identify mutant versions of these little worms that are defective in their development or behavior.
And those mutants can be maintained in culture in the laboratory in many cases. In fact, the mutants that we studied that led to this discovery are actually easy to propagate in the laboratory, even though they have lots and lots of developmental abnormalities. So it's a forgiving organism in terms of the ability to culture it in the laboratory and then, do genetic crosses and really work out mechanisms of genetic interactions that way.
Victor and his team were studying a particular mutation in these little worms called LIN4.
We were studying some mutations that affected C. elegans development in interesting ways, but, we were not expecting that what we found out would be the involvement of any sort of unexpected kind of molecular mechanisms.
At this point in the early 1990s, biologists already understood that proteins could regulate the expression of genes. They turn them on and off to help cells develop in what are called transcriptional programs.
We expected this would be a transcriptional program, controlling the sequences of developmental events that occur in the process of C. elegans development.
But instead, what they eventually found was a microRNA, a new type of biological molecule that turned on and off certain genes.
There was no reason to postulate, that there should be such regulators of gene expression. So this is one of those examples of where scientists are pursuing a question that's a very, let's say, central question in the field. And the expectations are that you're going to find out kind of more complexity, perhaps, and more nuance about mechanisms that we already know about. But again and again, this is just one example of when we do that, sometimes surprises emerge. And in fact, surprises emerge perhaps surprisingly often. So that's the nature of biology.
I'd love to know about the moment that you made the discovery that has led to this Nobel Prize. Can you tell me about that day when you realized?
In our lab, Rosalind Lee and Rhonda Feinbaum were working on this project. And Rosalind had been working on the project for several years because this was a very labor-intensive process. We were trying to track down a gene, and all we had to go by was a mutation, right? And so it was a gradual process of sort of homing in on the DNA sequence that contained the gene, that could function as a gene. And really the surprises started to emerge when we found that the pieces of DNA that were sufficient to confer the function of this gene were really small. And so that suggested, well, the gene is small, so the product of this gene is going to be pretty small. And then Rosalind worked to sort of pare down the sequence more and actually to mutate potential protein coding sequences in that little piece of DNA. And by process of elimination, she finally showed that there was no protein that could be expressed from this gene. And at the same time, we identified this very, very small transcript of only 22 nucleotides.
Nucleotides are the base of nucleic acids, the basic building blocks of ribonucleic acid, or RNA.
I would say there was probably, a period of a week or two there where these realizations came to the fore, and we knew we had something, new at that point.
Okay. And you mentioned Rosalind. She's your wife. Was she
Yep.
your wife at that point?
Yeah, actually, we've been together since 1976, and so, we started to work together in the mid-'80s. Yeah, and so we're still working together today.
Excellent. And she was the first author on that paper in Cell.
That's right. Yeah.
What's it like working together on a project like this and making those discoveries together, including with your wife?
Well, it's incredible. I mean, it's hard to express, and I'm sure you could try to imagine what it's like, to have this kind of validation, let's say, of our work. And the fact that we did it together, is just priceless. So,
do you feel like it's a Nobel Prize for her too?
I mean, yes. Every Nobel Prize has this obvious limitation of the number of people that they award it to, but of course behind that are the folks that worked in the lab, and the teams are always actually surprisingly large sometimes. That are actually behind the discoveries. In this case, 2 people in my lab, people in Gary Ruvkun's lab. So in a way, you can say they're the real heroes behind this. And our job, me and Gary, is to really represent this whole enterprise of science, which is so dependent upon teams, collaborations, brainstorming amongst multiple people, communications of ideas and crucial data. All this is part of the process that underlies a successful science like this.
What Victor and his team, including his wife Rosalind, had discovered was evidence of a new biological molecule called MicroRNA. It wasn't until 1999, though, 6 years later, that Gary Ruvkun's lab found a second microRNA called LET7.
Really, the watershed discovery in this narrative is when Ruvkun showed that LET7, the other microRNA, was actually conserved perfectly in sequence amongst all the bilaterian animals. So that meant that LET7 microRNA had been around for 500 million years. And so it was immediately obvious to the field that there had to be other microRNAs. This was not just a C. elegans thing, and it's not just LET7 and LIN4. There must be others. And that quickly emerged to be the case.
You mentioned Gary Ruvkun, and you'd been a postdoctoral fellow at the same time as him at MIT.
Yep.
But then at the point when you made your respective discoveries, you'd both set up labs. Would you call them rival labs? They were different labs and in the same town. I wondered.
I would certainly not call it rival labs.
No, okay.
Right, because we were working together as postdocs basically on this problem. We started to work on this problem of developmental timing in Bob Horvitz's lab, and I think we did at least one collaborative paper there, and then right after that, we were lucky enough to get a faculty position. And so we just basically informally divided up the work. And we anticipated that, there would be a point where we would get together and share information about what we've learned and see if we could come to a synthesis. And that was the plan. It was an informal plan. It was not really a collaboration. It was not really, certainly not rivalry. And actually that was a way of doing things that had been instilled, is that the word, on us from the culture of the C. elegans field at the time. I'm hoping you can imagine that there was an expectation in this community of C. elegans researchers that you should share data freely, And so far be it for Gary and me to start setting up rival labs,
Yeah.
Right?
No, okay. So you were talking on the phone quite often, were you? Chatting through your results.
Oh, we had, there was like a monthly Worm Breeders Gazette that was published out of the University of Minnesota, where you sent in the latest from your lab, unpublished work. And so it was just pre-internet. So there was lots of ways to communicate. We had local Boston area worm meetings, BAWM, which still goes on today.
Love it.
And folks showed up there and shared information, unpublished projects and stuff like that.
Your lab still works on microRNA. What are you thinking about now? What are you investigating? What questions do you still have?
Well, lately, one of the recent projects that I find very interesting is a project where we collaborated with a clinician, a geneticist who studies intellectual disability. And she had discovered that children with intellectual disability in certain families, carried a mutation that neither of their parents had. So they had acquired a private mutation, spontaneous mutation, in the protein that is associated with microRNAs, in humans. It's the Argonaute protein. And our genome contains 4 genes for Argonauts that are the partners of microRNAs.
So far, only 85 cases of Argonaute syndrome have been found in humans. This small gene mutation can have a range of physical and intellectual symptoms, including problems speaking and understanding language, delayed motor development, frequent seizures, cognitive impairment, and some autism spectrum disorders.
And so, working with these geneticists, our lab and other labs took those mutations that were essentially gifted to us by the patients. Right? And then we would put those mutations into our system. In our case, it would be into C. elegans Argonaute, the same mutation.
Victor's lab found that the process of gene silencing done by microRNA is disrupted by mutations associated with Argonaute syndromes.
What we've learned is that the mutant protein is sort of a rogue Argonaute, right? And it's basically screwing up the normal process that these 4 Argonauts usually do in us.
Other labs are doing similar studies, and a growing community of researchers is now meeting on an annual basis to share what they're learning about these mutations.
And the most exciting talks, in a way, were the talks by the folks who had taken the human mutations and then studied them in their experimental system. So I'm excited by this sort of a fascinating and amazing, essentially, collaboration between patients and their families and basic scientists, because those mutations are incredibly informative about the function of a protein that we have a lot of affection and fascination for.
Victor says the next step is to explore possible treatments.
This rogue Argonaute, in principle, could be removed from the system.
He says this could potentially be done using emerging technologies such as gene knockout, a genetic engineering technique that can turn off a particular gene, or through another technology under development called RNAi, in which RNA molecules work to turn off certain genes.
This is promising, and so I'm hopeful that the payoff for the patients will come.
Well, it's great that the original discovery, which led to all these new possibilities, has now got the recognition of a Nobel Prize. So congratulations once again.
Well, thank you very much. It's been fun.
It's not just Argonaute syndrome. Thanks to the discoveries of Victor Ambros and Gary Ruvkun back in the 1990s, medical researchers all over the world are now looking at how microRNA impacts the development of other diseases and potential treatments. I called up Justin Stebbing, Professor of Biomedical Sciences at Anglia Ruskin University in Cambridge in the UK, to find out more about the impact of microRNA's discovery in medical research.
MicroRNAs, like many processes, can go wrong. And they've been implicated in diseases as diverse as Alzheimer's and Parkinson's to cancer and kidney failure. So by understanding these really important molecules that turn on and off the 3 billion letters, that turn on and off our genes, we can understand the whole world of gene regulation, which is so important in so many different processes inside our bodies that it deservedly won the Nobel Prize this year in medicinal physiology.
And you're mainly focusing your research on cancer. How do you use and how have you studied microRNA in your own work?
One of the functions of microRNAs is they turn off genes. They stop certain genes being encoded and produced. And one of the groups of genes they turn off in cancer are called tumor suppressor genes. So they actually, in cancer, we found that they inhibit inhibitory messages. So you get a double negative effect. So the cancer's turned on more. And as cancers grow and spread, we find that they're implicated in every step of the way. To the extent that we can even find cancer early by measuring them in the blood or other fluid, and we've done that. And we can work out the right treatments for people based on what we call a microRNA signature. We can understand prognosis, which means how severe people's cancers are, but we can also try and harness them for treatments to make people better. We can directly target them. And microRNAs are now, for the first time, being considered as a drug in itself, because we can actually use microRNAs to turn off oncogenes, which are cancer-causing genes. So we can use it to what's called target those genes to make cancer less aggressive.
And how do you deliver microRNA in that way as a drug? Because does it not have to go really very precisely to the right damaged cell or the right part of the body? How does that happen, and where are the challenges in that?
That's a really, really brilliant question. And a lot of the field has been involved in that. If you remember, the 2023 Nobel Prize in medicine or physiology went for discovery of RNA as well. That's now being used in vaccines. Now, they're delivered via lipid nanoparticles. And so you can use those same little tiny lipid nanoparticles to encapsulate the microRNA and inject it into people. But you're right, it is a major subject of research. Some people believe we need to deliver it straight into the diseased tissue or the cancer itself via local injection. And other people are working on little cargo carriers, little nanoparticles that actually deliver it with a targeting molecule on its surface to an organ or to a tissue of interest.
Those scientists have yet to see a successful trial to deliver microRNAs in humans. Justin is confident that it's only a matter of time.
It's hugely exciting. The reason why a Nobel Prize was given is because it's massively expanded our understanding and comprehension of cellular processes and how genes are turned on and off, as well as all the other things, such as their valuable tools for studying gene function, cell biology, their promise as biomarkers, and we're developing microRNA-based therapeutics.
So what does it mean for biologists like you who work on and with microRNA that it's now got the international recognition of a Nobel Prize?
Advances in science is generally made in small steps, not giant leaps. The work which commenced with the discovery that Victor Ambros and Gary Ruvkun made in 1993 on a microRNA called Lin4 has taken 30 years to reach this point, and we're still working on it. I think it's given credibility to the area in that it's shown what people like me have believed all along, which is they're just such important molecules, and that without understanding them, we're really missing a large part of not just diseases, but how normal people grow. So this is taking it all to the next level.
Great. Well, thank you, Justin, for giving us a bit more kind of translational perspective on this discovery in a worm, essentially. So thank you.
Thank you so much for having me.
That's it for this week's episode. Thanks to Vivian Land from The Conversation US, who you heard at the top of the show, and to Clint Witchells at The Conversation in the UK, who worked with Justin Stebbing to help explain the significance of this Nobel Prize. The Conversations Global Network has been carrying reaction to all the 2024 Nobel Prizes on physics, chemistry, literature, peace, and economics. We'll put a link in our show notes to where you can read more of that coverage. This episode was produced by Katie Flood and me, Gemma Ware. Sound design was by Michelle Macklem, and our theme music is by Nita Saul. Stephen Kahn is our global executive editor. You can connect with us on Instagram at theconversation.com or email us directly at podcast@theconversation.com. Do sign up for The Conversation's free daily newsletter by clicking on the link in our show notes. The Conversation is a non-profit news outlet dedicated to sharing the work of academic experts with a wider audience. If you like what we do, please support us at donate.theconversation.com. And please rate and review the podcast wherever you follow us. Thanks for listening.